Hello judges of the Hackaday Prize 2015. With only a short time to demonstrate the virtues of my project, I'll have to keep things brief. So this is my entry, the DNA lamp diagnostic device. Although it's compact and cheap, this device has the potential to revolutionize medicine in the third world by bringing the very latest in molecular biology techniques to where it's needed, into the hands of the poorest 3 billion people on the planet. At its core, this device is a reactor and detector for the loop-mediated isothermal amplification of DNA, or LAMP for short. The list of diseases that LAMP can detect is very impressive, including humanity's biggest killers, such as HIV, tuberculosis and malaria. But with the right targeting sequences, LAMP can detect any sequence of DNA and RNA, and can even be used to detect GMOs in our food. So let's talk a little bit of science. PCR, or the polymerase chain reaction, is the key invention in molecular biology and can make billions of copies of the selected sequence of DNA taken from bacteria, humans, or samples of woolly mammoth found in the tundra. It's a pretty complicated reaction which relies on the splitting and joining of DNA helices and a polymerase enzyme that's taken from organisms that live in hot springs or hydrothermal vents, which copies the split strands. But that reaction has nothing on the complexity of the LAMP reaction. LAMP's reaction scheme is so much more complicated that I don't have really time to describe it in 5 minutes. But here are a couple of the key points worth mentioning. Firstly, it's basically just good for diagnostics. It doesn't make a single product that you can use in the lab. It just makes a huge variety of stuff made of multiple copies of the target sequence joined together. But, as it binds 6 to 8 distinct regions of the target DNA, it's much more accurate than PCR, which only binds 2. It can detect down to a few viral or bacterial particles. Secondly, as it doesn't need tricky PCR conditions, which require the rapid changes of precise temperature, LAMP is happy at a single stable temperature because the special polymerase has strand displacement activity, so we don't need to unwind the DNA with higher denaturing temperatures. Thirdly, with the discovery of viral polymerases from viruses that infect the single-celled organisms from hot springs, we now have access to polymerases that can process RNA and DNA, so now we can match sequences on viral genomes and detect things like HIV without additional work. Fourthly, so much DNA is made, things like the reaction mixture's pH and the amount of free magnesium in solution changes, so there are several ways to get a readout. Lastly, all the reagents and the enzyme can be freeze-dried, and are in this form stable at room temperature for many months. This project uses all these huge positive points and overcomes the 3 biggest hurdles in bringing this technology to the third world, which are: how do you take an advanced scientific technique and make it available to everyone, even people who can't read and write? How do you reach and maintain the precise temperatures required for this reaction in a place with barely any infrastructure? How do you make the readout obvious to the untrained eye without expensive equipment? And on top of this, how do you reach a price point that covers the more than 3 billion people who live on under $2.50 a day? I hope my device can be a part of the solution. Let's have a look at its features. It's super easy to use. You just add a small sample in a PCR tube which contains a pre-made mixture of reactants and push this button. Then a white light starts flashing, letting you know that the reaction is running. 15 minutes later, this RGB lights up with the diagnosis. Green means nothing was detected, blue means something was detected, and red means there was an error in the diagnostic procedure. That's pretty simple, right? It has polarity protection, and is designed to run on sources between 5 and 20 volts. The total cost of the device is about $10, but that could be cut in half if purchased in quantity. The cost for the individual tests, whether for malaria or viruses like HIV, is well under 5 cents. So, how does the device actually work? The device heats the sample and uses a tuned PID controller to keep it stable. It has a 405nm ultraviolet LED as an excitation source for fluorescent spectroscopy measurements, used to check for the activation of the calcine dye, which is caused by the release of magnesium in a positive reaction. What's there left to be done? Well, the PID code is the same as in my $5 DNA replicator, so that's sorted out. I've tested the fluorescent spectroscopy setup with fluorescent dyes and it works too. I've designed and fabricated the PCB boards and just finished populating it. So now what's left is getting the Arduino code running on the tiny 841 chip, 3D printing the light baffles and lid, and getting the aluminium heat block machined. You can keep track of my progress of my open source files on my project page. I hope you can support my project and wish me luck. Thanks.